This is Pseudomonas aeruginosa. It ranges from 50 to 60 micrometers long, but we're going to dive deeper into the DNA. For our experiment, we are only looking at one specific gene called PA14-70950, or BET-B. BET-B is found in between two other genes, BET-A and BET-I, as you can see in the figure above. BET-B consists of 1,475 base pairs, and we'll be using CRISPR technology to go in and knock down the whole BET-B gene to reduce the expression of it within the cell. You may be asking yourself, what is CRISPR? Well, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. While I'm not going to explain how it was discovered right now, I will tell you how we are going to utilize it. CRISPR is centered around a protein named Cas9, which binds to a small guide RNA sequence. This complex is then able to bind to the complementary sequence of the small guide RNA in the bacterial DNA. The Cas9 complex then cleaves the DNA at the binding site using endonucleases. This cleavage will deactivate the gene. For our experiment, though, we're going to use it a little differently, as we are using dead Cas9 or dCas9, which has deactivated endonucleases. So instead of cleaving the bacteria, the Cas9 small guide RNA complex will remain binded to the DNA, preventing expression of the gene. Once we have our knockdown strain of PA14-S0950, we can move on to our experiment to determine if the BET-B gene is significant for life. Since we determined BET-B is used to help the bacteria control osmotic pressure, we have created 3 environments, all with different osmotic pressures. The control wild-type strain and the mutant strain will be placed in the same exact environments. The first solution will be a 0.5 mole urea and LB solution, which will serve as an isotonic environment. Then there will be a 0.34 mole sodium chloride, which we anticipate will serve as our hypertonic solution. We will know if it is hypertonic if we see decreased cell growth in our control for this environment than when compared to the 0.5 mole urea and LB. Lastly, we will use deionized water as our hypotonic solution. Each solution will contain 500 microliters. 450 microliters of this will be the solution that we stated before, and 50 microliters will be the culture sample. The samples will then incubate overnight at 37 degrees Celsius. Once our samples have incubated overnight and have been collected, we will complete an 8-fold serodilution. By diluting our samples, it will make our results more accurate, as we will be able to clearly define each colony of bacteria that grows on the plate. We will plate our diluted samples and count the CFUs every 30 minutes over an 8-hour period.